Hello, everyone. Welcome to the quarterly Parkinson's webinar series brought to you by Cure Parkinson's, the Journal of Parkinson's Disease, and the University of Edinburgh. My name is Thilo Kunath. I'm from the Centre for Regenerative Medicine in Edinburgh, Scotland. And today we'll be discussing gene therapy for Parkinson's with a particular focus on neurotrophic factors. During this webinar, we have three great panelists: Professor Aideen Sullivan from University College Cork, Ireland. She's Chair of Pharmacology. Dr. Melanie Brandeber, a neurologist and vice president of clinical development at Capsida Biotherapeutics, and Tom Phipps, a Parkinson's research advocate and the first participant on the GDNF Bristol trial that you'll hear a lot about. We're going to do some brief introductions. There's a Q&A box that please submit your questions as we move along, and then we're going to get to these about halfway through the webinar. But we'll start with an introduction from Aideen first, please.
Hi, thanks, Thilo. So as Thilo said, my name is Aideen Sullivan. I'm a professor in the Department of Pharmacology and Therapeutics in University College Cork in Ireland. I'm a neuroscientist, and my research has focused on Parkinson's for almost 30 years. My particular interest is in the development of neuroprotective or disease-modifying therapies, which have the potential to slow down disease progression in Parkinson's. And in my laboratory group, we use preclinical models, both cells and animal models, to examine potential agents that might eventually be used clinically to help people with Parkinson's. My research group has a strong focus on neurotrophic factors as potential therapies, and of course, that's the subject of this webinar. One of our current projects in UCC, co-led by my colleague, Professor Gerard O'Keefe, is investigating two neurotrophic factors called GDNF and GDF5 in preclinical models of Parkinson's. This project is funded by Cure Parkinson's, and we're very grateful for their ongoing support. As well as leading a group of researchers, I'm also a lecturer in University College Cork. I teach neuroscience, pharmacology, and translational medicine. And I'm very keen that our science and medical students learn about the importance of communicating research to the public and to patients and their families. I'm a very strong advocate for patient and public engagement and involvement in research. And I involve people with Parkinson's in the co-design of my research projects at every opportunity. I have several ongoing research projects which have strong involvement in collaboration with people with Parkinson's. These include research on pesticide exposure and Parkinson's risk, various exercise strategies for people with Parkinson's, and most recently, a project on tracking symptoms and medications across the menstrual cycle in women with Parkinson's. Thank you.
Great, fantastic. Melanie, next, please.
I'm Melanie Brandeber, and I'm a neurologist, and I spent my clinical career taking care of people with Parkinson's disease and other movement disorders, and also serving as a researcher in Parkinson's disease and many, many clinical trials for Parkinson's drugs. So about 10 years ago, I moved to industry and was working on pediatric rare neurologic disease, mostly genetic disorders, and worked on drug development for 10 years. And then I had a chance to move to Capsida about 3 months ago, and was excited by this opportunity because they are working on an IV gene therapy for a subset, a genetic subtype of Parkinson's disease, as well as the opportunity to work on a gene therapy for a pediatric rare disease, STXBP1. So for me, this was an irresistible combination. Happily moved to Capsida and working on this therapy. The interesting thing about this therapy is that gene therapy has previously been very difficult to get into the brain. And typically, and in fact, some of my patients had this done, it was delivered by a surgical approach where the genes were actually put into the part of the brain most involved with Parkinson's disease. There were some issues with gene therapy because of the blood-brain barrier, which is very selective about what it lets into the brain. And we can discuss that a little more later. So the approach at Capsida is to use specially engineered capsids to bring gene therapy through the blood-brain barrier and into the brain. So this means you don't have to have a surgical approach. You don't have to inject anything into the spinal fluid around the brain. You get this therapy as an IV. So this is a very exciting opportunity. Has not been in the clinic yet. We are hoping to go into the clinic for clinical trials in 2025. So I'm very excited to be at Capsida and back in the world of Parkinson's disease, which I always loved, and very excited to take part.
Superb. Thank you, Melanie. Last but not least, Tom Phipps, please. Unmute, Tom, please. Almost there.
Right. Hello, everybody. Welcome wherever you are in the world. I'm very thrilled to be here. I'm Tom Phipps. I suffer with Parkinson's and have done since 2005. I'll try and slow down my speech because I tend to speak very fast. I hope you can hear me because it can be a bit difficult sometimes to understand. Now, I was diagnosed in 2005, as I said, and I went to raise some money a year after my wife had done a trek to Nepal for Parkinson's, and I did a cycle ride from London to Paris on the year of the Olympics, at around the time the Paralympics was starting in London. Started at Greenwich, got to Paris, got back in an absolute whirl of inspiration and energy. I was cycling like they would in the Tour de France at the end of 4 days. I was thrilled, and the first thing that our branch had was a meeting with Alan Woner, the director of the project, the GDNF trial. He gave evidence in the background of the previous trial, and then encouraged us to join the new trial. I couldn't see any problem with it. I investigated, and I just put my big size 9 boot, or 7.5 size boot actually, straight into the fold, and offered my chance to go as a research participant. A week later, I got phoned up by Lucy Mooney, the lead nurse on the trial, and she said, "What are you doing next week?" I said, "Well, I'm going to Bonfire Night." She said, "No, you're not. You're coming to join the trial." So I was taken on board for assessment, which involved a number of activities, and the rest is the history I'll talk about later.
Great. Thank you, Tom. So Tom's referring to this GDNF trial that took place in Bristol. That in the UK, a lot of you will know about it, but it will be explained in more detail as we're going forward. But we're going to start off nice and easy. Hopefully, we're going to try to define as many acronyms and technical terms as we can. So we're going to start with Aideen, and my question to you is, what is gene therapy, if you can define it in a lay way, and what is the difference between gene therapy and other emerging advanced therapy?
Sure. Okay, so gene therapy is a way of introducing specific genes into specific cells in order to treat a disease. So in Parkinson's, gene therapy is being used to introduce into the brain the genes for specific proteins that have the ability to protect dopamine neurons, those brain cells that are most affected in Parkinson's. And the class of proteins which are of the most current interest as an emerging therapy for Parkinson's are called neurotrophic factors. And neurotrophic factors are proteins that play really important roles in the brain. They help brain cells to develop normally in the embryo and in early childhood, and some of them also help to support and protect cells in the adult brain. And there are many different types of neurotrophic factor, and they each perform specific functions on different cell types. But there are a few that act particularly on dopamine neurons, and of course these are the ones that are of interest in Parkinson's therapy. And by administering dopamine neurotrophic factors to the brain of adults with Parkinson's, the hope is to kind of recreate the nurturing, supportive environment that is present in the brain during early development, which can hopefully protect the dopamine neurons from the ongoing degeneration and slow the disease progression. So the ultimate aim of administering neurotrophic factors then is to develop a disease-modifying therapy rather than simply reducing the symptoms, which is how most of the currently used Parkinson's drugs act. By using gene therapy to introduce these into the brain, it effectively means that the cells in the patient's brain would be able to make large amounts of these neurotrophic factors themselves. But another approach is to deliver the proteins themselves into the brain. This has a disadvantage compared to gene therapy in that the proteins would be quite rapidly broken down in the brain, and so their effects would be quite short-lived. So patients would need to receive regular injections of the protein to ensure a continuous supply to the brain cells. And most of the clinical trials to date on neurotrophic factors have used the protein delivery method, although some more recent ones have used a gene therapy approach, and we can speak about that later. But again, the main advantage of the gene therapy approach is to produce a longer lasting effect that can be achieved with protein delivery. And I'll just briefly mention one other emerging therapy, which is the use of cells, which can be transplanted into the brain in an attempt to replace the dopamine neurons that are lost in Parkinson's. So transplants of dopamine cells derived from fetal tissue and more recently from stem cells have shown great promise. But a major limiting factor, as mentioned earlier, when using either cells, proteins, or gene therapy is the need for invasive neurosurgery to deliver these agents to the brain, because the brain is protected by a structure called the blood-brain barrier, as was mentioned earlier. So at present, proteins, gene therapy, and cell therapies all need to be injected directly into the patient's brain using very precise and costly neurosurgical methods to target the specific area of the brain that's affected by Parkinson's. And this type of surgery can be associated with risks and side effects for the patient.
Fantastic, thank you. So if I'm going to summarize, the gene therapy is long-lasting. It's a therapy that can be delivered once, I think you said.
Yes.
It's usually delivered in a virus that Melanie will explain. While protein therapy is not long-lasting, protein needs to be administered multiple times, and Tom will speak about that. So the example that we're going to discuss is protein called GDNF. We use that term a lot. What it means is not very important, but I'll say it means glial cell-derived neurotrophic factor, GDNF. And it is a growth factor that specifically increases the health of dopamine neurons, which is why it's used. So GDNF Bristol trial is what we've been discussing, and Tom had the protein version of this trial. So Tom, what made you sign up for the trial in the first place? What were the challenges, and what was your experience of being on the GDNF protein trial in Bristol?
Well, one was stupidness, the impulsive nature of Parkinson's, which we're concerned about when we're looking at support for new trials. If you've got Parkinson's, you've got a cognitive chip that's damaged, but that's by the wayside. I was a scientist. I had a science degree. I was a primary school teacher, so I dropped away from science. But I still had that background of testing and understanding of research into biology and biological matters. And I made it my job to understand Parkinson's as much as I could. So what impressed me was the way that it had been backed up with sensible ideas for getting the blood-brain barrier past, a trustworthy, if you could call it that, delivery system, which involved a robot. And I'll show you my trophy. This is the perfect time. Here's my trophy I got from Renishaws, the designers of the system that went into this Bristol Head GDNF project. There's my title. It's a Tom Phipps, world #1 to have this port delivered into a system. And here's the machine that was programmed that took something like 20 hours. My operation took 20 hours. They've got it down to 4 hours now to get this pipe delivered into the brain, right through the skull, the top of my skull, into the putamen. And it was delivered by this robotic arm, which was programmed, as I said, as I was lying in the operating table. This is my head in a frame, and this is the port, just on the side here, that I've got in my head. Well, you can see that. Where's my camera? Yeah.
Can you show your port?
Yes, it is still in my head. And my son, when I have a drink, so that makes a nice game at the pub. Bum and I want another drink. Yes, so I thought, let's go for it. But the operation, as I said, took 20 hours. My wife said, why on earth is it taking so long? And they said, oh, the computer's slow. They had to go and find the program from somewhere else. I think he's left it in this car or something. And my wife said, oh, that's typical with him. He's always having trouble with his computer. And I came round and I was dreaming of carrots. I said, well, I want to go and see my allotment to get my carrots done. So it was a bit gung-ho, but I think I had every confidence that it was going to make a difference. I mean, it was a groundbreaking suggestion to put something straight into the brain like that. And the fact is that they're going to increase the number of neurons I got. It was a double-blind trial, so I didn't have the knowledge of whether I had the actual stuff going into my head. 4 tubes infused over an hour or so's period, sitting in a chair in a little porta cabin in Frenchay. And I wasn't allowed to speak to anybody about it, so we all sat there in silence. I could talk to the chap who was delivering it, but I couldn't speak to any of the other people. I was on my own anyway for most of the time. I had to go and have a CAT scan to check what the system was like, what the putamen was looking like, and how innovative it was. And that involved getting to Cardiff, being there half an hour after the plane had landed, with a delivery from Geneva of the radioactive isotope that was going to be put into my brain. That was put in, and I lay there for an hour and 20 minutes on a scanner. I was told to be still. How can you get a Parkinson's person to stay still? But I had to. It was purgatory. It wouldn't even let me play my iPad. So I got really cross with him. And well, after three scans, I had seen an improvement in my putamen, but unfortunately, it wasn't as much as it could be, because being the first one, my trial was ended abruptly because the whole thing got blocked. And they were able to see why it was blocked and change the design for future inputs. And that made a big difference to them, and it made the operation much slicker. And then subsequently to then, I've been talking to Renishaws and helping them design further kit. And that's how I got my pluck. Would you like any more of that?
Thank you, Tom. No, we'll come back to you in a minute, but that was excellent. Yeah. Melanie, after hearing all this and the challenges with protein therapy, what can you describe what the advantages of gene therapy are? But I'm going to ask you 2 sub-questions because you mentioned it. What is AAV, if you could describe AAV, and you use the word capsid. What is a capsid? So advantages of gene therapy, what is AAV, and what is a capsid? Your company's name is Capsida, so a capsida.
Yes, that's right.
Yeah. So,
that's right. So first of all, I would like to commend Tom on his bravery and pioneering spirit for participating in a clinical trial, especially one with an invasive procedure. It takes a lot of guts to do that, and that's how we move research forward. So I think that's terrific. I wanted to talk a little bit about gene therapy, the history of gene therapy, and the inherent problem of getting things into the brain. So the brain is very selective about what it lets in there. And things are generally gotten into the brain by a series of tiny capillary blood vessels, little tiny blood vessels, and things have to go across a series of membranes to get into neurons in the brain. And the brain is very selective about what it lets in. And if you think about it, you don't want everything you put in your mouth to go into your brain, right? You only want selective things going in there because that can cause problems. So typically, most IV gene therapy doesn't get into the brain very well. Gene therapy is generally done using a sort of a deactivated virus called an adeno-associated virus. And AAV9s are generally thought to get across the blood-brain barrier better than others, but still have pretty poor penetration typically, or historically. So what's been done in order to get genes into the brain are things like a surgical approach. So you get a hole similar to the one that they gave Tom, And then the surgeon inserts the gene into the part of the brain where they really want it to implant. But obviously that has potential side effects. You could potentially get infection, or as you go through the structures to get to where you want, you could potentially damage them. The other approach is to put it in the spinal fluid, the fluid that bathes the brain and spinal cord. And you can do that into the lower spinal canal, but then you're not going to get a whole lot of it up to the brain where you want it. So another approach is to put it in an area at the base of the brain called the cisterna magna, which is a kind of a nice big space to put something. But then again, there are some risks with that because you're right behind the brainstem, which is a critical area for things like breathing. So that's a potentially dangerous thing to do. So you have to have a very skilled neurosurgeon to do that. And then even if you put it there, it still goes in this fluid that bathes the brain, and you have to rely on it sort of diffusing into the brain. So the approach that Capsida is using is to try to take those AAV9 kind of disabled viruses and engineer them so that they are able to cross from those capillaries, those tiny blood vessels, into the brain tissue. And if you can do that, you can get really, really good penetration across the brain, because you're not just trying to get it into the spinal fluid or into a small part. You're actually getting it into every neuron potentially that is close to a capillary, and they all are, because that's how they get their food, right? So if you can engineer a capsid, which is sort of the AAV9 delivery system, to cross the blood-brain barrier selectively, you have a really good chance of getting into many, many more neurons than you do with other approaches. So what Capsida has done is they have a sort of soup to nuts operation that engineers and builds these capsids, these AAV9 molecules, to carry the cargo, the gene therapy cargo, across the blood-brain barrier in an efficient way. Now, the other problem with IV gene therapy is that you can have some toxicity associated with liver and other organs. So what Capsida has done is they've screened billions of different variants of these capsids to find ones that specifically go across the blood-brain barrier, but avoid going to the liver and other organs that you don't want targeted. So in our preclinical studies, it looks really clean and very, very effective in terms of penetration and very low in terms of toxicity. Obviously, it could be another thing when we go into patients, but we've done a lot of work to make sure that it's going to be safe and effective in patients. And we're very excited to go into the clinic next year for an IV therapy, for gene therapy for a particular subtype of Parkinson's, GBA, Parkinson's disease. Did that cover everything?
Yeah, can I ask you?
Needed?
Yes, that was great. I wanted to ask you a specific question, and I wanted to make a description of a capsid, and I want to ask if you think it's correct. So could I describe a capsid as particular proteins that decorate the surface of viruses?
It's sort of the hollowed out shell of the virus is the way I think of it. And you can see the little drawing up next to me. Oh, it's the other way. It's kind of a picture of that. You've basically got a shell, and you are carrying a cargo. This is kind of a simplistic way to think about it, but this is the way I think about it as a non-chemistry major, is that you are using this kind of protein shell to transport the virus and deliver it into the brain. And these viruses they've been modified, so they're not infectious and they can't replicate themselves. They can't make more copies. They are just specifically these kind of shells to transmit the gene into the brain.
And one other question about intravenous delivery, so putting the virus into a vein, and you said the capsid would allow it to pass the blood-brain barrier because
Yes.
of the outside part of it. But then does it go everywhere in the brain, or can it be also further targeted to areas affected by Parkinson's?
It goes everywhere in the brain, but it does have very good penetration into the parts of the brain involved with Parkinson's. The striatum that Tom mentioned and the basal ganglia, that part of the brain. But some forms of Parkinson's that are genetic affect more neurons than just the ones deep in the brain that we typically associate with Parkinson's. So we think it's going to affect a lot of maybe some other symptoms of Parkinson's potentially.
Great, thank you. Thank you, that explains that. So Aideen, I'm gonna bring you back in just to give us a picture of the landscape of the current neurotrophic factors being trialed, whether protein or gene therapy, and what may be some of the emerging ones.
Yeah, absolutely. So to date, most of the neurotrophic factor trials in Parkinson's have been on GDNF and the related factor nurturin, which is very similar to GDNF. And there was very, very strong evidence from many years of preclinical research that each of these neurotrophic factors could protect dopamine neurons from degeneration, improve their function, stimulate new growth, and even regenerate them in animal models in the lab. And the early clinical trials using both GDNF and nurturin, were very successful. But unfortunately, the later, more kind of trials with larger numbers, longer placebo-controlled trials like the Bristol trial did not meet their endpoints, so were deemed to have failed. So there's still a lot of work to be done in this space. But just briefly, GDNF was the one that's been used the most, and that was delivered via a protein approach. So the direct protein itself was delivered, whereas nurturin was delivered using gene therapy. So in the GDNF trials, as Tom explained earlier, patients would need to come back every 4 weeks, typically, to have a kind of a top up of the GDNF in the port, as Tom described. The nurturin trial was a one-off delivery because it used gene therapy for a long-lasting approach. And it is worth saying that there is a new clinical trial that's recruiting at the moment called the Regenerate PD, which is using GDNF using an AAV gene therapy approach, and that's just been announced in the last couple of months. So that's kind of the state where we are at the moment with the trials to date. But I suppose you asked about emerging factors, and just to mention about those, just I suppose the scientific thinking behind looking for other factors, if you like. I suppose the preclinical studies and early stage trials didn't really translate to the larger placebo-controlled trials. And there are many reasons for this lack of success, or many possible reasons, including the delivery methods, the patient selection. A lot of focus has been on trial design. For example, the length of the trial was quite short, and a lot of people think the trials should run for longer to see meaningful results, and the outcome measures that were used to determine the outcomes. There's a lot of questions about these, but some researchers have suggested that one reason for the lack of translation to the clinic of GDNF and GDF5 might be to do with some changes that happen in the brains of people with Parkinson's. So specifically, it may be that proteins called RET receptors, which are essential to enable GDNF and nurturin to have protective effects on dopamine neurons, they might be decreased in the brains of Parkinson's because of the pathology related to alpha-synuclein overexpression in these brains. So this reduction in RET receptors might result in the growth factors have been less effective than they were in the preclinical work. And this has prompted researchers to test alternatives, so-called RET-independent factors. One of these is called CDNF, and this has been extensively investigated by Professor Mart Saarma and his colleagues in Finland. And CDNF works on dopamine neurons in quite a different way to GDNF and nurturin. It actually works on organelles inside the cell, the endoplasmic reticulum, to reduce stress mechanisms and protect the cells from within. So that's quite different from the other factors tested to date. And there's quite strong, very strong preclinical data in animal models of Parkinson's of GDNF. And then following on, that was recently tested in a phase 1 clinical trial using the protein delivery approach. And this was a safety trial showing that CDNF was well tolerated by patients and safe, and some positive effects were seen in some patients. However, that approach is not going to be continued. The company have decided that they're going to try and develop this agent in a smaller form, which can cross the blood-brain barrier and then be delivered through, they mentioned a subcutaneous or intranasal route. So that's kind of underway at the moment. So that's one alternative factor. In our group in Cork, we've worked for many years on another RET-independent neurotrophic factor called GDF5, and this is the work that's funded by Cure Parkinson's, I mentioned earlier. And we have found in over many studies preclinically that this has strong protective effects on dopamine neurons. It's a member of the same family as GDNF, so it's quite related, but it works through a different receptor, not the RET receptor. And we found that the receptors that GDF5 needs to work on dopamine neurons are very well maintained in the brains of people with Parkinson's. So this is something we're investigating as an alternative. I think one possibility might be that using factors that work through different pathways or different mechanisms could be ultimately be used in combination to produce better and more effective therapies, but that's under investigation at the moment.
Great, thank you. If I can summarize. So GDNF has been trialed several times at the protein levels, such as the Bristol trial, but there has been other trials. Early success in phase 1, maybe even phase 2, but placebo-controlled trials failed to meet primary endpoint. Nurturin, very related to GDNF, was tried via a gene therapy, AAV Nurturin, that was delivered to the brain. It wasn't delivered via IV, and that also failed to meet its primary endpoint. But both of these, all of these trials, the trial design may have been problematic or it wasn't long enough. So the third factor you mentioned was CDNF, and this is really coming out of Helsinki in Finland, and this is quite a different neurotrophic factor, but that is showing some promise, and it's being delivered via protein, I believe, at the moment. And a new version of CDNF that's a bit shorter, that crosses the blood-brain barrier, is also being trialed. And then the fourth factor you mentioned, GDF5, is that correct? You're working on in Cork, and this is an interesting factor because it doesn't go through the RET receptor, which is how GDNF works, but the RET receptor may be reduced in Parkinson's, but the way GDF5 works is through a different mechanism that it could be more effective in Parkinson's.
Exactly.
I think that was the summary. Yeah. So I'm going to move to Tom now, and Tom, I want to hear a bit more about the practical challenges that you had during the GDNF protein trial, and just from what you've been listening, what do you think some of the advantages you might see with gene therapies that are emerging? So unmute, Tom, please.
Thank you. I was thinking about the actual negative response to the Beat GDNF trial, what the negatives were, because I think we learned from that. And it covers your point. The trial involved a lot of subjective assessment of the condition of the person that takes part. There were assessments on their speed of response. There were assessments on their mental state, and of course, being in part of a trial, your mental state is heightened, and you become much more aware of that. But it's also very subjective. And the other issue is that I think the trial involved questions and sets of tasks that an ordinary person could actually use, if they do it regularly, can actually improve their performance, and they practice at home and they can get the whole thing becomes better. So it's not ideal having a subjective assessment like that, because it does cause complaints, difficulties in validating the results. However, I'm going to look on the positive side of things and say that I think I was getting the drug because things happened in my body, and I just think I've had a positive response to it long term. And that's another issue. They didn't carry the research on long term. They didn't look at us in a long term situation. I finished early because my system, the port filled up, blocked at the top, and because there are 3 silicon bung for each particular tube, and the needle goes through it, and the needle was sharp, and it abrased some of the silicon, and that slewed off and got stuck in the tube and blocked the tube. So that failed. So about 50% of my tubes, 4 tubes, got blocked, or 2 got blocked, and the others were struggling, and it just didn't deliver the stuff. I had 9 months of it. It was about 9 months before it started to block. That first 9 months, I had 9 months of GDNF, fortunately, so I had the advantage. and the following 9 months I had very little. So I didn't see the major the differences that other people got. Going forward, the test has actually got some positives. The positive thing was we had a protein that would work, that was improving despite being stuck on a trolley in a scanner for an hour and 40 minutes. It was quite heartening to see the Putamen increasing in response, and that was excellent. So that was worthwhile doing, and it's actually spurned a lot of other CDNF and the GDFR and even the viral factor. The other thing is that the tubes were delivered in my brain vertically to the putamen, and that got in the wrong aspect to get into the putamen. The putamen lies in that plane, and it was coming in from the top. So imagine it. So it's only delivering to a small part of my putamen. So they've decided to come in from the rear, back of the skull. So the patient was lying down on their head, on their front, and the delivery was put from the rear, and it got into the putamen in a much more agreeable way. The other issue is there's a reflux on the tube, which Renishaw's were able to adjust to allow the spread of the condition, the gel to go, the GDF to go wider, and that was an innovative decision they made. Be that as it may, I would never give up on a trial. I always take the chance to go on a trial if there's one that's worth it. I can't do it anymore because I've had this thing I've done already, but I think it's worth doing. Anything else you want to know? Sorry.
What are your thoughts on gene therapy?
I think it's the way forward. I think one dose and your brain starts to sort itself out. Because if the genes can be improved, get rid of that gene that's been transformed allegedly from pesticides in our environment. I can't think of any other reason why the proliferation of Parkinson's, the number one neurological condition, should be it for any other reason than the environment, because there wasn't there before Parkinson in the same numbers it is now. And they had, in the Victorian times, they had awful conditions, very bad atmosphere. I was brought up in the Midlands, and the atmosphere there was pretty heavy with coal dust. And not until about my early teens did it clear up. By then I was in Weston-super-Mare, breathing lovely fresh sea air. But the only thing is, I went to the allotment with my dad every week and got a dose of paraquat and all sorts of stuff. DDT put on the allotment. He just couldn't be bothered to weed. He just used the weed killer to do it. Thank
you, Tom. Yeah, that's certainly. There's a lot of evidence around pesticide use and increasing Parkinson's in the environment. But good to hear that you think gene therapy is the way forward. Yeah. Melanie, we're going to come back to you because you have alluded to a gene therapy that is in your pipeline. So it's not a neurotrophic factor, and you mentioned subset of Parkinson's. But can you explain a bit more about the gene therapy that you are pursuing?
Sure. So we are trying to treat for our first Parkinson's study with our capsids. We're trying to treat a subset of Parkinson's due to a mutation in the GBA gene. So those of you who are familiar, and I've seen some questions coming through about various gene subtypes, is that the trend now is to do gene subtyping in Parkinson's, not happening everywhere, but sort of becoming more prevalent. And we're finding out that some Parkinson's cases are caused by genes, mutated genes of various types. And so, there's LRRK2, there's PARK1, et cetera, and one of them is GBA. And so what GBA mutation causes a decrease in glucose cerebrosidase, which is an enzyme involved in lipid metabolism. So the theory is that if you disrupt your lipid metabolism, you are causing a sort of a chain reaction of things in the brain, including probably impaired processing of alpha-synuclein. And as many of you know, alpha-synuclein that is misfolded is a key factor in the development of Lewy bodies. And Lewy bodies are kind of the pathologic hallmark of Parkinson's in the brain. And so this may lead, along with a lot of other things that we probably don't completely understand, because we never really do in science, probably contributes to neurodegeneration in various parts of the brain, including the parts involved with Parkinson's symptoms. So our approach is to put a good gene in that makes glucose cerebrosidase, or we call it GCase for short, which is a lot easier, so that hopefully this GCase will be able to restore appropriate lipid metabolism and decrease the this chain of events that we think leads to Parkinsonism. So we would be using a subset of patients, but this is a pretty large number of patients. We think this is about 10 or 15% of patients with Parkinson's disease have this gene. So this could be thousands and thousands of patients with Parkinson's due to a GCase deficiency. And what we see in those types of patients is they may have, their symptoms may present earlier, progress more rapidly, potentially have some cognitive dysfunction, some thinking and memory problems, maybe earlier than they would have gotten it with other types of Parkinson's. So we are hoping to go in and hopefully see a good clinical response when we get into the clinic next year.
Great, thank you, Melanie. So we're going to move to Q&A. There's questions piling in, so we'll see how many we can get through. Melanie, I'm going to come back to you because there's someone interested in the blood-brain barrier and asked a question. Can you work with the gut microbiome to deliver a therapy?
I would say the gut microbiome is a super hot topic. In fact, I know of someone who's doing a study just in the GBA subtype on the microbiome. Really, really interesting work. I'm not familiar with work going on specifically with a therapeutic approach there, but I'm sure there will be at some point. And at this point, we're not in a position to give gene therapy in association with the gut microbiome.
Great, thank you. So Aideen, I'm gonna ask a very direct question. What is your thinking? What is the thinking about success moving forward between cell therapy and gene therapy?
That's a difficult question, and I honestly think both have real promise. There's a really exciting trial going on at the moment in Europe. Multi-center trials, Sweden and UK are leading this, called the STEM-PD trial, and this is really based on decades of work using dopamine transplants. And this particular trial is using dopamine neurons made from stem cells, and it's so many top researchers have been working on this for such a long time, and I really think that this is going to be successful, and that I think is in the future. But I don't think we should give up on neurotrophic factors. I don't think we should give up on all of the work we're talking about today. Again, there's a wealth of evidence there that these factors can work, that many patients have responded really well, even in those trials that have been deemed not to be successful. But there are factors that have to be explored about, as I mentioned earlier, trial design, trial length, getting the right delivery mechanisms, as Melanie and others are working on. So I think both approaches have a really promising future.
Great, thank you. Yeah, I'm of the same opinion as well. Tom, this is not quite related to gene therapy, but you mentioned about pesticides. What do you think about diet? How do you think diet could maybe affect Parkinson's, improve, or maybe promote it?
Well, I think it's very important, because the whole thing is tied in together. It's the whole, how you look after your body, keep yourself fit and healthy. I am suffering, I think, from adoration of sugar. I'm taking on too much sugar, and I think it's affecting my sleep, my awareness at night, my memory, my skin condition, and it's all these 6 points that they say towards keeping your body healthy. As a result of taking too much sugar, so I'm going to have to reduce my sugar intake quite considerably. But the thing is, I have found that if I keep my gut healthy, keep my bowels open, for want of a better word, it makes a big difference to the levodopa as its absorption through the gut. If there's a blockage, it stops it going in, and if tablets go in and they get the stomach attacking them, well, that's even worse. So the sooner we move away from medication like that is better. However, I don't think you should forget about your body. Keep fit, keep healthy, because you give yourself a better chance.
Yeah, really good advice. Really good advice. Melanie, I'm going to ask you about a question from Leslie. Is gene therapy likely to help patients who have not responded well to dopamine delivery mechanisms such as pills or patches?
That is a very good question, and I'd say we're sort of early on in knowing what gene therapy could do for Parkinson's. If there has been a lot of neuronal damage and death, we obviously we're not in a position at this time to regenerate cells that have died or been seriously injured. But I think there's a lot of hope. I would also like to say that, I would like to echo what Aideen said about the importance of working on a variety of different therapies, because gene therapy isn't going to be right for every single patient based on stage of disease or type of symptoms or immunologic issues, because you're going to have to take probably at least some corticosteroids if you get gene therapy. So there are issues that I don't want to come across that gene therapy is the absolute answer to everything, because I don't think it is. It's really important that we move forward sort of spherically on developing therapies for Parkinson's.
Yeah, great. Aideen, I'm going to ask you the very difficult question. It's coming in from two people. So how close do you think we are to adopting a gene therapy generally for people? Which is a very difficult question, and time frames are always problematic to predict. And a very similar one, what time frame for gene therapies to become available to patients? How long do you think a cure will be found? So these are very tough questions. So Aideen, sorry to put them on you.
That's okay. It's almost impossible to answer. So gene therapies are being used in some patients in clinical trials right now, and the numbers are very small, of course, but we are at the clinic, if you like. They are being administered to patients, but in terms of rolling out to all patients, there are always going to be huge challenges with that. As Melanie said, these type of approaches, either stem cells or gene therapies, are not going to be suitable for all patients. So there are limitations, I suppose, to the rollout to all patients. But I suppose the answer is they're in patients now. They've been well tolerated, and some really promising results being seen with the gene therapy approach from the Nurturing trials. There has been really nice post-mortem data from patients 10 years after the Nurturing trials showing sprouting and regrowth of those dopamine neurons. So the future is bright, but whether they will be rolled out to all patients, I think that's just impossible to answer. It also depends, of course, on companies, deciding to invest in these and rolling them out. So not really within my expertise, I'm afraid, to talk about the kind of the company side of it or the funding side.
Can I ask, for people interested in keeping up with the progress of gene therapy, cell therapies, or even the protein therapies, what is the best advice for someone living in the US or the UK? How can they keep up with it and just know what's on the horizon?
Are you asking me, Tilo?
Yes, yeah, maybe UK side or
for the UK
then,
side, I mean the Cure Parkinson's website and Simon Stotts, is really great on social media as the Science of Parkinson's. He's got all the updates. As soon as a paper is published, it's on Simon's social media. So I think that's a really good place to just watch for anything that's happening in Parkinson's, this topic and others. The Parkinson's UK website is also really, really good and up to date. And then in the US, well, Melanie might want to speak, but the Michael J. Fox website is excellent too, and they have funded, of course, some of the gene therapy and neurotrophic factor trials in the US. So to me, those are the go-to websites and social media sites for up-to-date information.
Yeah, and Michael J. Fox also has a trial finder tool. So I completely agree with you. Michael J. Fox Foundation, Parkinson's UK, Cure Parkinson's, keeping up with the latest information is so important because lots of things are happening, lots of trials are happening. Melanie, do you have any advice for the people asking about time frames for gene therapy, etc.?
I completely agree with looking to your local patient organization and the Michael J. Fox website and other good things. If you want to see specific clinical trials in your area, you can look at clinicaltrials.gov, and that will show you kind of what research is being done. And while, you know, being in a clinical trial isn't for everybody for a lot of different reasons, it's a very brave and sort of selfless thing to do. And that's how we make progress, because we can do everything we want in a lab or with animals or whatever, or, you know, but there's nothing better to test a drug than to put it into a patient, a brave patient like Tom, who's willing to give it a shot.
Yeah, I was going to come to Tom next. So Tom, can you give some advice about advocacy and what have you been doing since your experience with the trial to let other people with Parkinson's know about what it is like to participate in a trial?
Well, I go and give talks wherever I'm asked. I write a blog every month in our branch. I've been elected president for the last 2 years of the local branch, so I put my 5 pence in, which is surprising. I have lots to say. But the other thing is, I remember the trial. One thing positive that's come out of it. There's the scheme that they use for installing my drug is being used for chemotherapy for children, and it's brilliant. And I think that is a positive thing to take. If you take a trial, you're doing some good. But the other thing is, I'm a step on the way to a cure. It won't be in my lifetime, but it's on its way. So I think every time you do it, it's a positive thing.
That was beautifully said. Thank you, Tom. I really appreciate that. So a difficult question about can gene therapy be used to treat dystonia? I think I saw that question somewhere. I don't know if that's for Aideen.
I haven't come across that as a treatment for dystonia, to be honest with you. Melanie might know more.
I don't know that there's a specific therapy going on. Dystonia also has many different causes, some of which are genetic and some of which are structural, et cetera. So I suspect what will happen is there will be a therapy developed for a particular type of dystonia, maybe a genetic form of dystonia, and that if that's successful, it could potentially be applied to other forms of dystonia.
Thank you. Peter has an interesting question. I'm going to rephrase it a little bit. Can genetics be used to identify the type of Parkinson's you have? And could that inform the type of neurotrophic therapy you get, or even the delivery, if you know what kind of genetic form of Parkinson's you have? And maybe Melanie, I can bring that back to you.
Yeah, and I think I would want Aideen to chime in as well. But yeah, potentially, again, depending on what exactly is the word we use is pathophysiology, like what is happening between the gene and the symptoms to cause Parkinson's, could potentially inform us as to what neurotrophic factor might be more helpful. And Aideen, if you could chime in on that, I'd appreciate it.
Yeah, absolutely. There hasn't been any specific work on this, but just in theory, I suppose, you know, the more we know about the type of Parkinson's a patient has, the more we can tailor therapies. So I think in some of the questions earlier, people talked about specific, like maybe the PINK1 mutation and different mutations like that, which are very common amongst the genetic forms of Parkinson's. This is not really relevant to neurotrophic factors themselves, but, you know, in terms of gene therapy as an approach, I guess we can see perhaps those being targets down the line for specific subtypes. But at the moment, it's not being kind of tailored to those types of Parkinson's as far as I know.
Great, thank you. We're almost near the end. I'm going to ask Tom for any final thoughts before I wrap up.
Right, okay. Just about lasting out. Thing is, don't give up Parkinson's. there's every chance you can live a reasonable life if you give yourself a chance. I've been out on the allotment picking apples today. I spent the first hour in the morning getting myself ready. I had to go and have my crown fitted in the dentist, so it took a while, and I've come on to this. So I've managed a whole day working. I shall discover tomorrow that I'm absolutely worn out, but go for it, guys. And it's worth it going for the cure, but there is ways of living with Parkinson's.
Great, thank you, thank you. So we're going to do one more question, and then I'm going to do a sum up. So there are specific therapies. Are there specific therapies being trialed for LRRK2, LRRK2, which is a genetic form of Parkinson's? GBA seems to be winning the popularity contest. So maybe Melanie can speak about LRRK2 versus GBA.
I have not looked recently to see what's happening in LRRK2. So, but again, if you look on the Michael J. Fox website or clinicaltrials.gov, you can find out. I think LRRK2 actually was more popular than GBA for a while there in terms of research dollars. So I would be willing to bet there are some LRRK2 studies going on. I just haven't looked recently.
Yeah, that's true. There's huge amounts of LRRK2 activity in the UK, especially in Dundee, in small molecules especially. So LRRK2 is an enzyme, and you can find drugs that inhibit this enzyme. So I think there is still lots of efforts on LRRK2. So it's not losing any popularity contest.
It's not.
Okay, so I'm going to try to summarize what we've heard today. So gene therapy is a type of advanced therapy that can deliver genes to the brain, usually in a virus, a very safe virus. And the surface of that virus is important for whether you need to inject it into the brain directly or through the capsid route intravenously. And the things that are being looked at to be delivered are neurotrophic factors. And these are factors that can increase the function of existing neurons, prevent neurons from dying. They have lots of great properties in labs and in the preclinical studies. But importantly, a lot of human studies have already been trialed. So we don't know how far it is away generally, but there's certainly lots of human studies that have been conducted and are being conducted. So the point that Aideen made is that the phase 3 trials for GDNF, Nurturin have not met their clinical endpoints, meaning they weren't successful. But the trial design, the length of the trial, there could have been a number of issues of why they didn't meet the clinical endpoint. It doesn't mean that the therapy wasn't working, but it could have just been the trial design. So there's certainly no reason to give up hope on GDNF and all of these other factors. So there's even more growth factors in the pipeline. So CDNF was mentioned. So CDNF is a completely different type of molecule. It sounds the same as GDNF, but it works differently. But it does increase neuronal survival. And it was mentioned that maybe combining CDNF and GDNF could be a combination therapy. So GDF5 being worked on actively in Cork has not yet been trialed in humans, but the data in preclinical models looks excellent, and it has a different type of mechanism than GDNF, so it has lots of promise. So I just wanted to also say, recap some of what Tom said. So Tom has been mentioned a number of times, have been a hero, brave, a patient, being patient number 1 in the GDNF protein trial in Bristol. And so much was learned from his experience, from the people running the trial and moving forward. And as he says, he's been a critical factor in the wheel of finding a cure for Parkinson's, and even unexpected advantages like finding a way to deliver medicine to children with gliomas has been learned from Tom's experience. So I think and comments that Tom has made, I think we should really take on board and encourage people to sign up for trials when they can. And I think the last thing is that is to be active with your local groups. Try to learn as much as you can about what's going on. Everyone always wants to know, when is a cure going to happen? How long are these things going to be generally available, not just for a trial, but for the healthcare system? And I think the best way to keep up to date with that is through the amazing work that charities in the UK and the US and worldwide are doing. So Michael J. Fox was mentioned already. They're doing incredible work in the US, well, globally. They're doing incredible work globally. But Cure Parkinson's and Parkinson's UK, I think these three organizations, there's more, I'm sure, but these three organizations are doing fantastic work. And please sign up to their newsletters and get as much information as you can. Okay, that's it for the webinar. Take care, everyone, and we'll see you hopefully in 3 months' time. Thank you. Bye-bye.